Hello everyone, my name is Brian Xu. Today I'm going to talk about fast-growing trees because in recent years, many news are talking about deforestation and I think that the fast-growing trees can help to rebuild forests. That's why I choose this topic to research. Hope you will like my presentation. First of all, what is a GM tree? GM refers as the genetically modified tree, which is a tree that the DNA has been modified using genetic engineering techniques. It can be done by manipulating the cells, which helps the tree to grow faster. So, why is it important? Well, due to the extreme climate changes, many trees had died because of the environment are not able to support trees to survive. For instance, when drought comes, trees will die because there is no water for them. People also cut trees for wood products and land, which increase the deforestation in the earth. This graph shows that the carbon dioxide in the air has increased rapidly after the Industrial Revolution. If people do not find a solution for it, the CO2 in the air will keep rising, which will cause the ocean temperature to increase. When ocean temperature rises, the sea level will also rise because of the melting of ice in North and South Pole. Because of this, planting fast-growing trees can help us to reduce the amount of CO2 in the air that can reduce the effects of global warming. That's why many scientists are researching for the method to grow trees faster. What are the PXY and CLE genes? PXY is a receptor that coordinated cell divisions in the procambium. It controls both of the orientation and the rate of cell division in the procambial stem cells. Increasing the amount of CLE41 results in more cell divisions that increase sizes, which with well-ordered vascular development. Study shows that manipulating PXY-CLE signaling pathway increases vascular cell divisions in embryophysis, will increase the size of the trees and make it grow faster. The first step of the method is to make promoters. The first promoter, 35S promoter, go through gene cloning. The second promoter, SUC2, generated by PCR, will increase the cell division of the trees. Now, I will explain the process of gene cloning with the animation I make. The hollow orange circle represents plasmids while the scissor represents restriction enzymes, and the other one represents foreign DNA strand that was cut by restriction enzymes. First, the restriction enzymes cut through the plasmid. The foreign DNA then will come and connect to the plasmid. After that, the plasmid will be put into the bacterium. The bacterium begins to reproduce, and later, the foreign DNA will be transferred into plant cells by bacteria. Without bacteria, the DNA strand will not be able to be transferred into the plant cells. PCR is also an essential reaction used in fast-growing trees, and these experience overlap. PCR is used to make SUC2 promoter. The number 3 and 5 on the animation symbolize the prime of the DNA strand. There are 2 DNA strands in this process. I made one strand blue and another green to prevent confusion. So, if 2 different DNA strands want to join, they must have the overlap part in order to connect. The small blue and green strand on the animation is the primer. When the temperature rises to 90 degrees, the DNA will denature like this. After the temperature dropped to 68 degrees, the top green strand 3' to 5' read from the left will anneal to the 5' to 3' blue strand that is also read from the left. The 2 strands will then connect by bases. red circle represents the tech polymers. The polymers will come and extend the DNA strand when temperature rises to 72 degrees. Now we have a 4 strand of DNA combining 2 different strands. The result of the previous animation is shown in the orange strand of DNA. When temperature rises to 92 degrees, the strand will denature again. When the temperature drops to 56 degrees, the primer will then attach to the strand. The tech polymers will elongate the strand in 72 degrees. This is a method of overlap PCR. Results. The University of Manchester found out that using PTT-PXY and PTT-CLE's 41 genes are the best way to increase the tree growth. This picture of this page and the next one shows the different promoter they use to test the experience. It also shows how the PXY and CLE work to increase cell division, improve air quality, prevent soil erosion, and produce better quality woods are some advantages of a fast-growing tree. If the CO2 in the air can be reduced like this graph, we will be able to prevent the temperature of the ocean to rise. Companies can also cut these trees to make profits, which prevent deforestation if they do not cut down so rapidly. However, the trees can also affect native plants in the forest that might cause forest fire when the trees are grown out of control. I think that investing and planting fast-growing trees is necessary because people are now facing some serious problems such as global warming. Trees can help us to prevent these problems from getting more and more serious. And this is a bibliography of the presentation. Thank you very much.